Assalamu alaikum wa rahmatullahi wa barakatuh. I'm teacher Ahmed Jamul. I'm glad to talk about document 5 of chapter 1, oogenesis. Back in document 1 of this chapter, you have learned about the female's reproductive system. What you see in the figure is a front view of this system. In the vagina, which is the site of sperm deposit, you can detect a huge number of sperm cells that will move up the female's genital tract heading towards the fallopian tube. During their movement in the female's genital tract, capacitation of sperms occurs. What does that mean? It means that the sperm cells acquire their capacity to fertilize within the female's body. But not all these sperm cells manage to reach the fallopian tube, and most are lost on their way. When in the fallopian tube, the sperms meet the one female gamete produced by the ovary. When one sperm cell penetrates this female gamete, fertilization occurs, and all other sperm cells degenerate. You must be asking, now, how come females produce one gamete while males produce millions of gametes? Is this the only difference between the production of gametes in males and in females? That's why in this document you are going to learn about gamete production in females, a process that is also referred to as oogenesis. You will get to know how does oogenesis in females differ than spermatogenesis in males. By the end of this video, please recheck the following objectives to make sure that you covered them all. Document D of your books shows the process of oogenesis that occurs in the ovary. Just like spermatogenesis, oogenesis occurs in 4 phases. During the multiplication phase, the mother germ cell called oogonium multiplies. But what is different in females is that this phase occurs during their embryonic life, which means that the oogonia multiply inside the ovary of the female's embryo. The second phase of oogenesis, which is the growth phase that is equivalent to an interphase, occurs also during the embryonic life of the female. Just as the third phase, which is meiosis, is about to start, oogenesis stops at birth of the female. The cell at this stage is oocyte 1, blocked at prophase 1 of meiosis 1. Few oocytes will only resume meiosis once every month, starting from puberty. An oocyte 1 that undergoes the reductional division gives 2 haploid cells with N chromosomes. One large cell called oocyte 2 and another very small cell that is called the first polar body. This polar body will soon degenerate, maybe before or after it continue its meiotic division. But the oocyte 2 will undergo its second meiotic division, and just as this oocyte 2 reaches metaphase 2, it will be released from the ovary by ovulation, and meiosis will be arrested for the second time at metaphase 2. Now, if fertilization does not occur, meiosis will never be resumed and oocyte 2 will survive for 24 hours only, and thus oogenesis won't be completed. But if only this oocyte is fertilized by a sperm cell in the fallopian tube, meiosis will resume again and the oocyte 2 will release its second polar body. And so an ootid is formed, and this would be the differentiation phase. The ootid nucleus will then fuse with the nucleus of the sperm to give the zygote. As for the state of chromosomes in each of the germline cells during oogenesis, it is similar to that in spermatogenesis. An oogonium has 2 N chromosomes with 1 chromatid each. An oocyte 1 has 2 N chromosomes with 2 chromatids each. An oocyte 2 has N chromosomes with 2 chromatids each, while an ootid has n chromosomes with one chromatid each. Now, you must have noticed that an ovary usually produces one oocyte by ovulation only once per month, while a male daily ejects millions of sperms. That's why an oocyte is never found alone inside an ovary. It is well protected by a structure called the follicle. Document A, page 27, shows a cross-section of the ovary in which you can observe the different developmental stages of these follicles with oocytes inside them. You can see very small primordial follicles made of the oocyte surrounded by few follicular cells. These follicular cells multiply and the follicle grows bigger into a primary follicle. As more layers of follicular cells are added, the follicle becomes secondary follicle that house the oocyte 1 blocked at prophase 1. The follicular cells secrete a fluid that accumulates in a cavity inside the follicle. At this point, the follicle is known as the cavitary or tertiary follicle. This tertiary follicle will continue to grow in size until it becomes a mature follicle ready to burst and push the oocyte 2 outside towards the pavilion of the fallopian tube. This mature follicle is called the Graafian follicle. Then the ruptured follicle will transform into a functional structure called corpus luteum or yellow body that continues its transformation into a non-functional structure called the corpus albicans or white body. Document B of your books shows the structure of this Graafian follicle. It shows the oocyte in the middle of the follicular cavity still blocked at prophase 1. This oocyte is surrounded by a non-cellular protein layer called the zona pellucida. This zona pellucida is in turn surrounded by a cellular layer known as corona radiata. Corona radiata is the innermost layer of the cells that directly surround the oocyte both before and after ovulation. Those cells make the cumulus oophorus. Now, surrounding the follicular cavity that is filled with the fluid are many layers of follicular cells or granulosa. In addition to other cellular layers are called theca interna and theca externa. The development of the follicle from primordial follicle to Graafian follicle is known as folliculogenesis. Document C of your books shows the different stages of folliculogenesis. About 500 primordial follicles which have been at rest around 10 years resume their growth activity before each menstrual cycle. 3 to 4 months later, only one follicle will reach maturity, the pre-ovulatory follicle, while the others degenerate. After being developing for 4 months, the follicle in the ovary has a diameter of 5 mm. At this point, the growth of the follicle becomes accelerated so that the follicle grows to become with a diameter of 20 mm within an average of 14 days only. Only a few hours after the formation of the Graafian follicle, this follicle ruptures to ovulate the oocyte 2 into the fallopian tube. The ruptured follicle in the ovary turns into a luteal body or yellow body that degenerates after 14 days. Once the yellow body in the ovary degenerates, the female experiences menses, which is the bleeding discharged from her uterus. Menses marks the beginning of a new cycle where the events shown in the figure are repeated again. The cycle has an average duration of 28 days, where ovulation occurs on day 14. The duration before ovulation is known as the follicular phase because of the presence of mature follicle in the ovary, while the duration after ovulation is called the luteal phase because of the presence of the luteal body. The duration of the luteal phase is always fixed in a female 14 days, while that of the follicular phase varies from one female to another. So you can see in a female we cannot talk about oogenesis without talking about folliculogenesis at the same time because they are two synchronized processes. Before birth, oogonia multiply until reaching birth, the period where multiplication stops, and the growth phase begins where oogonia become oocytes 1 blocked at prophase 1 of the first meiotic division. Focus on the development of the follicles starting from birth till puberty. Then, 4 months before the first menses, where the primary follicle develops into cavitary follicle. At this point, which is day 0, the female cycle starts by the follicular phase, where the cavitary follicle develops into the Graafian follicle, which will be at day 14 ready to rupture by ovulation and release oocyte 2 blocked at metaphase 2. After ovulation, the oocyte 2 might die if not fertilized within 24 hours of ovulation, and so the cycle enters the luteal phase where the ruptured Graafian follicle continues its development into yellow body, then into white non-functional body that will degenerate at day 28, indicating the end of the cycle. But if the oocyte 2 in the fallopian tube gets fertilized by a sperm and it resumes its second meiotic division and releases the second polar body, then forming a fertilized ovum that develops into an embryo. On the other hand, the yellow body persists in the ovary for 3 consecutive months during pregnancy that transforms into white body and degenerate. Now, do you remember document F of document 1 of this chapter? I guess now you will be able to understand this document really good. Back then, you have learned that the genital activity is cyclic, means that it happens every 28 days. This activity is interrupted when a pregnancy occurs for 9 months, and the genital activity is finally arrested at an early elderly age when all the follicles found in the ovary of a female at birth are depleted almost by the age of 50. And that's how we come to an end of this chapter. The whole life cycle of human being alters between a diploid phase, which starts at fertilization, and a haploid phase, which follows meiosis, that occurs in the reproductive system of each male and female. Hope this video was helpful. Please read part 5 of the summing up, solve probing the document, and the following parts of probing document 6.